Welcome to Literary Insights. This is the summary of the book, Brain for Innovation, The Neuroscience of Imagination and Abstract Thinking. The book examines why humans are uniquely innovative through the lens of neuroscience. While imagination is not uniquely human, humans have a greater capacity for abstract thinking. Abstract thinking allows for innovations involving conceptual knowledge and is key to major technological advances. The book argues this capacity for high-level abstraction is what truly distinguishes human innovation. It explores the neural basis of imagination and abstract thinking. Part 1 focuses on the hippocampus and imagination. Part 2 examines the neural circuits underlying imagination. Part 3 analyzes abstract thinking and the prefrontal cortex. The book also discusses how language, creativity, and cultural evolution have fueled innovation. It aims to understand the neural foundations that have enabled humans to collectively advance knowledge and technology on a global scale. The hippocampus plays a critical role in memory formation and encoding new memories, as demonstrated by the famous case of patient HM, who underwent medial temporal lobe surgery, including removal of the hippocampus. HM developed severe anterograde amnesia, unable to form new memories after the surgery. He also had retrograde amnesia, affecting recent memories more than distant ones. This showed that the medial temporal lobe, including the hippocampus, is necessary for encoding new memories as well as recalling recent past memories. Older memories remained intact. The temporally graded retrograde amnesia supported the theory of memory consolidation. New memories are first encoded in the hippocampus and then consolidated to be stored in the neocortex long-term. Having two separate memory storage sites, hippocampus and neocortex, is advantageous. Hippocampus allows remembering details to aid decision-making, while consolidation in neocortex frees up hippocampus for encoding new memories continuously. Later research in 2000s would discover that the hippocampus is also involved not just in memory, but imagination of future events as well, challenging the view that its role is only for memory. The hippocampus plays a role in both temporary memory storage and permanent memory storage. It can temporarily store detailed episodic memories while extracting general facts and storing them as semantic memories in the neocortex over time. There are two main memory storage sites: the hippocampus for temporary storage of experience details and the neocortex for permanent storage of the gist general facts extracted from experiences. Memory consolidation involves actively selecting valuable memories from past experiences and recombining them through imagination rather than just passively storing incidents. This allows for innovation by applying past lessons to new situations. If you liked the book, you can purchase it using the links in the description below. By buying through these links, you contribute to the channel without paying any extra, as we receive a small commission. This helps us bring more quality content to you. Damage to the hippocampus impairs both memory formation and vivid imagination, showing its role in both functions. Brain imaging also shows hippocampal activation during remembering past events and imagining future ones. The hippocampus is part of the default mode network, which is activated during internal mentation like daydreaming, thinking of past, future, and creative problem solving when unengaged with external tasks. This relates imagination and memory functions in the hippocampus. In summary, the passage discusses the dual role of the hippocampus in temporary memory storage and permanent memory consolidation through extraction of general facts, as well as its involvement in both memory and imagination functions through its role in the default mode network. The passage discusses how memory of an event can differ from what actually occurred. This is because the brain processes and stores memories differently than a computer. Some key points. Memories tend to extract the gist meaning rather than precise details. Information can get mixed up over time. Missing details may be unconsciously filled in by the brain based on expectations and recent experiences, making eyewitness testimony unreliable. False memories of entirely fabricated events that never happened can form, as shown in some famous cases like the George Franklin and Paul Ingram cases. Suggestions from authorities can potentially implant false memories. Elizabeth Loftus conducted experiments demonstrating how misinformation can alter true memories and even lead people to form entirely false memories of events that never occurred, like being lost in a shopping mall as a child. Subsequent studies have replicated these findings on false memory implantation using methods like introducing a fake visual image to subjects. In summary, human memory is fallible and can differ significantly from actual events due to various cognitive factors. Studies using electrode implants in rats found that hippocampal neurons called place cells fire when the rat is in a specific location. This indicates the hippocampus creates a cognitive map of space. Remarkably, when rats rested or slept, the same sequences of place cell firing would replay, as if re-experiencing a past experience. This phenomenon is called hippocampal replay. Replays provided insights into how the hippocampus is involved in both memory and imagination. The reactivation of neural patterns from the past suggests the hippocampus supports remembering previous events. Replays also indicate the hippocampus can simulate sequences not actually experienced, supporting a role in imagination. Animal studies allowed direct observation and manipulation of hippocampal neurons to uncover neural mechanisms like replay that help explain the hippocampus's role in memory and imagination. While invasive in animals, such studies provide insights not possible from non-invasive human research. So in summary, place cells and hippocampal replay demonstrated through animal studies help explain how the same neural circuits in the hippocampus can underlie both remembering the past and imagining possible future events. Here is a summary of the key points. Matt Wilson developed a new parallel tetrode recording technique that allowed simultaneous recording of up to 100 hippocampal neurons in freely behaving rats. This was a major advancement that enabled the study of real-time neural network dynamics. Wilson discovered that the sequential firing of hippocampal place cells during active navigation is replayed during subsequent REM sleep. This supported the hypothesis that dreaming facilitates memory consolidation. Later studies found that place cell sequences are also replayed during sharp wave ripples that occur during slow wave sleep and awake quiet rest periods. Replays happen much faster than real time. Replays are not just repetitions of experienced sequences, but can include novel, unexperienced trajectories. This suggests replays may support memory retrieval, planning for future navigation, and imagination mentation beyond just consolidation. Wilson's discoveries set a new trend in researching the dynamics of place cells and how the hippocampus represents both spatial trajectories and temporal sequences of events through replays. Researchers reconstructed a rat's movement trajectory based on the sequential firing of place cells in the rat's hippocampus during a sharp wave ripple event, while the rat was sitting quietly on a maze. Importantly, the reconstructed trajectory did not match any route the rat had actually traveled on the maze. This suggests that hippocampal replay during sharp wave ripples can represent both experienced and unexperienced potential spatial trajectories. This study demonstrated that hippocampal replay is not just a reactivation of past experiences, but may also involve imagination of potential future experiences or routes. This fits with evidence that the hippocampus plays a role in imagination in both rats and humans. The findings showed that two independent lines of research in humans and rats arrived at the same conclusion, that the hippocampus is involved not only in memory, but also in imagination. Here is a summary of the key points about the medial temporal lobe and hippocampus. The medial temporal lobe includes structures like the hippocampus, entorhinal cortex, perirhinal cortex, and parahippocampal cortex. The hippocampus is involved in memory formation and consolidation. It receives input from the entorhinal cortex and sends output to other brain regions. Santiago Ramon Cajal's drawings in the late 19th century provided early insights into the structure of the hippocampus. His drawings showed subregions like the dentate gyrus, CA3 and CA1, the circuit involving the dentate gyrus projecting to CA3 and then CA3 projecting to CA1 is known as the hippocampal trisynaptic circuit. This has been a major focus of research on hippocampal memory functions. CA3 has highly recurrent collaterals that allow its neurons to strongly excite each other. This enables CA3 to maintain activity patterns in the absence of external input. David Marr's influential theory proposed that the CA3 recurrent collaterals allow it to store memory by strengthening connections between coactive neurons, forming cell assemblies that can regenerate the original activity pattern. This is known as content-addressable memory. Later research also found the hippocampus stores sequences or patterns that unfold over time. The recurrent collaterals in CA3 may also support sequence memory and memory consolidation over time. In summary, the hippocampus and medial temporal lobe, especially the CA3 region, are critically involved in memory formation and consolidation via its neural circuit architecture and highly recurrent connections. CA3 is thought to play a critical role in storing and retrieving experienced event sequences, episodic memory, and imagining unexperienced event sequences. This is because CA3 neurons are massively interconnected through recurrent projections, allowing partial activation of neurons to sequentially activate others. Sharp wave ripples, during which most hippocampal replays are detected, are initiated in CA3. This provides evidence that CA3 is important for storing and retrieving experienced sequences and imagining unexperienced ones. CA1 is different from CA3 in that it lacks strong recurrent projections. However, CA3 and CA1 neurons show similar place-specific firing patterns, suggesting they process similar spatial information. It is theorized that CA1 selects and reinforces high-value sequences generated by CA3. This suggests CA1 may play a role in value-based processing and decision-making. David Marr made foundational contributions to understanding the hippocampus and computational neuroscience in general through his influential proposals about the hippocampus, neocortex, and cerebellum. His work on the hippocampus laid the groundwork for ongoing research. Dopaminergic neural systems play a role in neurological and mental disorders like schizophrenia, Parkinson's disease, and addiction. Dopamine neurons in the midbrain change their firing in response to reward delivery and cues predicting rewards. Some dopamine neurons specifically signal reward prediction error, the difference between actual and expected rewards. Studies found that the activity of dopamine neurons correlates with reward prediction error. Regions encode the value of rewards. The hippocampus was also found to represent value signals. Rats were trained to choose between targets with different water reward probabilities. Recordings found that neurons in the CA1 region of the hippocampus encoded the changing values of the reward probabilities at the different targets. This was surprising since the hippocampus was thought to mainly represent spatial and cognitive signals, not value. But numerous brain areas, including the hippocampus, represent reward value in decision-making. So in summary, the passage discusses evidence that dopamine neurons encode reward prediction error, and many brain regions, including the hippocampus, represent the value of rewards, playing a role in reinforcement learning and value-based decision-making. Rats were trained in a task where they had to choose between two targets to get a water reward. The reward probabilities of the targets unpredictably changed over time. To maximize rewards, the rats had to use reinforcement learning to estimate the evolving reward values probabilities of the two targets based on their past choices and outcomes. A reinforcement learning model could predict the rats' choice behavior, suggesting they were updating target values trial by trial. Neurons in the hippocampal region, CA1, but not the subiculum, showed activity correlated with the trial-by-trial value estimates from the reinforcement learning model. This was surprising. Further experiments found much weaker value signals in CA3, the main input to CA1. This suggests CA1, not CA3, is representing the value signals. Inactivating just the CA1 region impaired the rat's ability to learn and update values from outcomes, affecting their choices. Inactivating other hippocampal subregions did not. These findings indicate CA1 has a special role in representing value information that is important for its functions, unlike other hippocampal subregions. Here is a summary of the key points from the chapter. The simulation selection model proposes that the hippocampus simulates and reinforces high-value events and actions in preparation for the future, rather than just remembering the past. It suggests this function is implemented in the CA3-CA1 network of the hippocampus. CA3 is proposed to act as a simulator, generating diverse event sequences based on its massive recurrent connections. It simulates both experienced and unexperienced sequences. CA1 is proposed to act as a value-dependent selector. It preferentially reinforces high-value sequences generated by CA3 based on neural activity related to value. This allows neural representations of high-value sequences to be strengthened, guiding optimal choices in the future. Evidence suggests CA3 replays are generated independently of value, while CA1 replays are preferentially shaped by value. The dentate gyrus may play a role in binding diverse sensory signals to form spatial context, but its exact role in the simulation selection process requires more research. The hippocampus is thought to perform simulation selection of spatial trajectories through replay and value-dependent activity in CA1. This allows an animal to plan and choose optimal routes between locations. The ultimate evolutionary reason for this function is unclear. One question is whether birds, which can fly directly between places, need this ability. Studies on the bird hippocampus are limited compared to rodents. However, comparative analysis across species can provide insights into conserved and varied mechanisms. The mammalian hippocampus, including humans, has a surprisingly similar gross anatomical structure across species, with distinguishable DG, CA3, and CA1 regions. In contrast, the hippocampus of non-mammalian vertebrates like fish, reptiles, and birds does not show the same clear anatomical divisions. This suggests the mammalian hippocampus structure evolved for simulation selection abilities. So, in summary, comparative studies indicate the mammalian hippocampus structure supports simulation selection functions, while birds may not need this due to different mobility. More research is still needed, especially on the avian hippocampus. Birds and mammals both have better spatial memory compared to cold-blooded vertebrates like fish and reptiles. However, the avian and mammalian hippocampi have distinct anatomical structures. Studies implanting microelectrodes in pigeon hippocampi found goal cells active at rewarding locations and path cells along paths between rewards. This differs from rodent place cells, which fire regardless of reward locations. It is proposed that the need to navigate between arbitrary locations on land, facing obstacles, selected for the hippocampal simulation selection function in mammals to model optimal trajectories. Birds can fly directly to goals without such need. Whale hippocampi are much smaller, suggesting degeneration of spatial function without land navigation demands. Bat hippocampi are well developed with place cells, perhaps due to specialized maneuvering in enclosed spaces rather than straight flight paths. So in summary, the evolution of hippocampal simulation selection for modeling spatial trajectories is argued to have occurred specifically in land-navigating mammals due to their navigational challenges, not in birds or whales who lack such demands. Bats may be an exception due to their specialized flight environments. The hippocampus is involved in imagination and simulation of potential future events and trajectories, not just spatial navigation. This capability for imagination is likely shared by many mammals. However, human innovation and ability for abstract thought goes beyond just imagination. It involves the ability to derive general concepts and principles from individual examples through abstract thinking. Abstract thinking is not unique to humans. Many animals can recognize patterns and represent general rules to maximize survival. Neural evidence also shows abstract representation of categories, relationships, rules, and social cognition in other animals. Even at the neural level, some level of abstraction is inevitable as neural networks store overlapping and distributed memories. Common features of similar memories will become associated, representing more abstract concepts. While abstraction exists in other animals, humans have an exceptional capacity for high-level abstraction using complex concepts unrelated to direct physical experiences. This superior ability for abstract thought, when combined with the basic capacity for imagination, is what truly distinguishes human innovation and creativity. The neocortex plays a key role in supporting this high-level abstraction. Neural networks can develop generalization, a form of abstraction, through experience. Neurons repeatedly activated by similar stimuli will strengthen their synapses, allowing them to respond to novel but related stimuli as well. This explains how we can recognize novel objects as belonging to a familiar category. Kant proposed the concept of innate abstraction, that humans are born with innate cognitive principles independent of experience. Neuroscientific evidence supports this in grid cells in the rat entorhinal cortex, which represent space using a hexagonal grid pattern that is consistent across environments. This suggests an a priori representation of space. The medial and lateral divisions of the entorhinal cortex carry abstract structural knowledge and sensory experiences, respectively, feeding into the hippocampus. This parallels Kant's view that we understand the world through an interplay of experience and innate concepts. Humans have a much greater capacity for abstract thought than other animals due to our advanced language ability and ability to routinely think across concrete and abstract domains. Category mistakes reveal our innate tendency to consider abstract concepts and entities as if they were concrete, showing abstract thought is seamlessly integrated into our cognition. A larger human brain size allows for higher processing power, supporting abstract thought. But it is the distribution of neurons, particularly in the neocortex, that truly distinguishes human intelligence and abstract ability. Neuron count varies significantly across animal species. Elephants have more cerebellum neurons than humans, while humans have more neurons in the cerebral cortex than any other animal. The expansion of the neocortex, especially in humans, likely drove our advanced cognitive abilities. The neocortex makes up 80% of the human brain, compared to just 1% in rats. Primates have a higher neuronal packing density in the cerebral cortex compared to other mammals, allowing for more cortical neurons. Humans also have the largest brains of all primates. The rapid expansion of the human neocortex over the last 2 to 3 million years, through neuronal circuit duplication rather than new circuit invention, is a key factor in human cognitive specialization. While the hippocampus plays a role in memory and imagination, the content is dictated by information from the neocortex. The neocortex expansion thus enabled more advanced imagination and abstract thought in humans. The human capacity for high-level abstract thinking and innovation is likely due to the expansion of the neocortex during brain evolution. One clue for understanding the neural basis of abstract thought comes from examining the prefrontal cortex, as it is implicated in executive functions like planning, reasoning, and cognitive flexibility. The prefrontal cortex is most developed in primates, especially humans, suggesting it plays an important role in human unique cognitive abilities. The case of Phineas Gage, who had damage to his prefrontal cortex, showed his personality and decision-making abilities were altered, indicating the prefrontal cortex is important for flexible, goal-directed behavior. To be innovative, one needs both persistence towards long-term goals as well as cognitive flexibility, abilities linked to the functions of the prefrontal cortex. Examining this brain region provides clues about the neural underpinnings of human abstract thought and creativity. The prefrontal cortex is important for maintaining persistence towards long-term goals and flexibility in changing behavior based on environmental changes or hidden rules. Damage to the prefrontal cortex can cause issues with distraction, impulse control, planning, and adapting behavior appropriately. This is demonstrated using examples like the Wisconsin card sorting test. The prefrontal cortex represents abstract concepts needed to achieve goals, like task rules. Neurological studies show activity related to abstraction even in animal prefrontal cortices. The human prefrontal cortex likely supports even higher-level abstract thinking due to its larger size and evolutionary importance for social behavior. Within the prefrontal cortex, the frontopolar cortex may play a key role in abstraction, as imaging shows more anterior regions tied to abstract thinking. Around 40,000 to 50,000 years ago, modern human behavior emerged characterized by innovations like language, art, trade that demonstrated more advanced abstract thinking abilities. This human revolution may have been enabled by brain changes supporting higher-level abstraction. Archaeological findings from the Upper Paleolithic era, such as cave paintings, figurines, and elaborate burials, suggest that early modern humans had advanced cognitive abilities comparable to today, including symbolic and abstract thinking. It was previously believed that anatomically modern humans emerged around 40,000 to 50,000 years ago in Europe. But fossil evidence now indicates Homo sapiens emerged much earlier, around 200,000 to 300,000 years ago in Africa. There is a gap between the emergence of anatomically modern humans and evidence for fully modern human behavior. The cause of this transition is debated. Some argue a genetic change increased cognitive capacity, while others argue environmental and social factors drove changes. More evidence is still needed to fully understand when and how modern human behavior and cognition emerged. Paleoneurology, the study of endocasts, may provide insights into evolutionary changes in brain structure. But detecting subtle changes is difficult, and the fossil record is incomplete. Here is a summary of the key changes in the size and shape of the brain case during human evolution. Brain shape became more globular, rounded between 100,000 to 35,000 years ago in Homo sapiens, matching the shape of modern human brains. Earlier Homo sapiens fossils from 300,000 to 100,000 years ago did not have this modern globular shape. The parietal cortex and cerebellum regions of the brain bulged out more, contributing to the increasingly globular shape. Within the parietal cortex, the precuneus area expanded disproportionately relative to chimpanzees. The precuneus size is also correlated with parietal bulging in modern humans. The precuneus expansion in particular has been proposed to be associated with recent human cognitive specialization, since the precuneus is involved in various advanced cognitive functions like abstract thinking, creativity, memory, and self-awareness. While brain size was similar across groups, it was the changes in shape, especially the parietal cortex and precuneus bulging, that distinguished modern human brains and correlated with new behaviors in the archaeological record around 100,000 to 35,000 years ago. In summary, the brain case became rounder, and certain regions like the parietal cortex and precuneus expanded more markedly in Homo sapiens between 100,000 to 35,000 years ago, correlating with modern human behaviors and cognition. This points to important neurological changes underlying human cognitive evolution. Deep learning refers to learning performed by a multi-layer artificial neural network. Having more hidden layers allows the network to perform more complex computations and learn hierarchies of features. Early experiments found that adding more hidden layers improved performance on image classification tasks, sparking the deep learning revolution beginning in 2012. Hidden layers learn to represent features at different levels of abstraction, from low-level edges to complex, class-specific patterns. This enables visual object recognition. Unsupervised learning experiments found that deep neural networks can learn high-level abstract features, like faces, without explicit labels, just from visual inputs. Some studies found networks develop properties like number coding or face selectivity spontaneously without any training, suggesting depth enables innate abstraction. The human brain's greater depth of cortical connections compared to other species may enable its strong abstract reasoning capabilities. Areas like the prefrontal cortex and hippocampus deal with high-level abstract concepts due to their high-order positions in neural networks. Neurons in the hippocampus and medial temporal lobe of epileptic human patients showed diverse responses to different visual stimuli. Remarkably, some neurons responded selectively to specific people or objects, like one neuron that responded to pictures and names of actor Jackie Chan, but not other people. These concept cells responded to abstract concepts like personal identity, not just visual features. Attempts to find similar cells in animals have been unsuccessful. The human hippocampus may handle particularly high-level abstract concepts, though other interpretations are possible. It has direct communication with the prefrontal cortex, a region involved in abstract thought. Expressing the human ARHGA11B gene, important for neocortex development, in mice, ferrets, and marmosets, increased neocortical size and enhanced flexibility. But expressing it in marmosets was halted before birth for ethical reasons. The findings suggest the neural basis of human abstract thought is unclear, but being directly studied, though with ethical concerns, to better understand differences from other animals. Language is a uniquely human ability that elevated human collective creativity to a new level. It is universal across all human societies. Key features of human language include its discrete units, phonemes, words, grammatical rules, productivity in generating new sentences, and ability to refer to non-present things. While some animal communication shows limited features of language, no animal system fully displays all the properties of human language. Attempts to teach language like sign language to great apes like Coco have been controversial. Skeptics argue the apes may just be associating behaviors with rewards rather than truly using language creatively. Their language skills do not surpass a young child's. The exact areas involved in human language processing in the brain are still being studied. But neuroscience research suggests language emerges from complex interactions between brain regions rather than being localized to one single area. Language is a key part of human evolution and what separates humans from other species. While language is essential for sharing ideas and accumulating knowledge across generations, studying the neural basis of human language is limited due to the lack of a good animal model. Nevertheless, two key brain regions involved in language processing have been identified: Broca's area and Wernicke's area. Damage to Broca's area results in non-fluent aphasia, while damage to Wernicke's area results in fluent aphasia. The classical Wernicke-Geschwin model of these language areas is an oversimplification, and newer models focus on finer-grained cognitive processes. There is debate around whether language shapes thought or is merely a tool for communication. While thought is possible without language, language likely influences abstract thinking through its role in representing concepts. Both views, that language enhances thought and that it is mainly a communication tool, have support. In summary, while progress has been made in identifying brain areas involved in language, fully understanding the neural basis is limited without animal models. There is also ongoing debate around the relationship between language and thought. Language plays an important role in representing abstract concepts like liberty and integrity, not just concrete concepts like table and cat. Language is highly abstract. People with aphasia, language impairment, show little impairment in abstract reasoning, arguing against the view that language enhances abstract thinking. However, language deprivation in childhood can lead to deficits in cognitive functions like understanding cause and effect, and abstract thinking skills like chess, math, feelings, social boundaries. This suggests language may play a role in developing abstract thinking during childhood, even if it's not strictly necessary for abstraction. Language could help scaffold and refine abstract thinking during critical periods. Understanding the origin of human language is challenging due to lack of direct evidence and animal comparisons. Theories include a sudden genetic mutation versus gradual evolution over time. FOXP2 gene mutations were thought to potentially explain a sudden language emergence, but evidence now suggests changes occurred earlier in human evolution before splitting from Neanderthals. Most researchers now favor a gradual evolution of language abilities over a long period through early naming of objects' actions and increasing grammar complexity over time. For early hominins to develop language, they would need the ability to encode sounds into discrete speech units like consonants and vowels, rather than just undifferentiated calls. This would require changes in how the brain controls vocal tract muscles and interprets sounds. Stringing individual words together into simple messages with meaning was an important step, though not yet as complex as modern language. This type of rudimentary proto-language is seen in young children and language learners. Further developments added grammar elements like plural markers, tense, clauses, allowing for richer linguistic structure. There are debates around whether language evolved from other cognitive abilities or was a standalone development. Theories propose links to abilities like shared intentionality, information exchange, or application of recursive thinking to communication. Developing language also required establishing high levels of social trust through cultural practices like religion and ritual, according to some coevolution theories, as words alone are unreliable signals. Mirror neurons in the premotor cortex that activate from both performing and observing actions provide a neurological basis for the mirror neuron hypothesis of language origins. Creativity refers to generating ideas that are both novel, original, and useful, effective. It involves imagination, but imagination alone does not guarantee creativity. There are two modes of creativity. Deliberate creativity involves focused reasoning to solve problems and is associated with the prefrontal cortex. Spontaneous creativity involves generating ideas in a relaxed state or during rest sleep and is associated with the default mode network. In reality, creativity involves a dynamic interplay between controlled deliberate processes and spontaneous processes. Neural networks involved in creativity include the default mode network for spontaneous idea generation and the central executive network including the prefrontal cortex for evaluation and meeting task goals. Dynamic interactions between the default mode network and central executive network are observed during creative tasks, suggesting creativity emerges from an interplay between unconstrained ideation and constrained evaluation selection. While imagination supports creativity by generating novel ideas, imagination alone during unconstrained states like rest-sleep may not necessarily yield high-value creative ideas, as imagination is value-neutral. Effective creativity requires an additional evaluation step. In summary, the passage discusses two modes of creativity, the neural networks involved, and how creativity emerges from an interplay between spontaneous imagination, ideation, and deliberate evaluation selection, rather than from imagination alone. Here is a summary of the key points about spontaneous creativity from the passage. Spontaneous creativity occurs during idle states when the default mode network is activated. This allows the hippocampus to generate diverse activity sequences through weakening of inhibitory control. The content of imagination is constrained by and related to past memories, as the hippocampus retrieves and recombines previous experiences. To enhance spontaneous creativity, one should expose themselves to a broad range of experiences so the hippocampus has more diverse material to generate ideas from. Breadth of knowledge and interests widens the scope of imagination. Factors like open-mindedness, embracing diversity of ideas, and tolerating failures also promote creativity at both the individual and organizational level. Relaxing the mind through relaxing activities like sleep, bathing, or travel can promote spontaneous creativity by freeing thoughts from deliberate thinking and exposing the brain to novel stimuli. Alternating between focused work, flow, on a problem, and relaxed states helps ready the brain to generate related ideas spontaneously by shaping neural connections during focus periods. The passage discusses the topic of human innovation and its future implications from both optimistic and pessimistic perspectives. From a pessimistic view, human innovation has enabled the destruction of ecosystems through overexploitation of resources, habitat loss, and climate change. Earth is undergoing a sixth mass extinction event due to human activities, with current extinction rates being hundreds to thousands of times higher than the background rate. If these trends continue, they could eventually lead to the extinction of humanity as well. The optimistic view believes that future innovation will address these problems by developing renewable resources, more sustainable practices, and technologies to curb climate change and restore biodiversity. Innovations in fields like AI, biotechnology, and space could create new solutions to global issues and improve human welfare. However, the optimistic view also acknowledges risks from advanced technologies that must be properly managed. Overall, the future consequences of innovation remain uncertain and depend on how humanity chooses to guide technological and societal progress. Both opportunities and challenges lie ahead if current destructive trends continue versus if innovation is steered towards sustainability. Predicting which path emerges is difficult, but important for influencing outcomes. Ecological diversity can be increased by forcing existing species to evolve into new species through environmental changes. However, if the changes are too abrupt or drastic, it can lead to mass extinction, as most living species may not be able to adapt quickly enough. Historically, catastrophic events like large volcanic eruptions and asteroid impacts have induced drastic environmental changes, killing many species. The most severe mass extinction about 252 million years ago wiped out over 70% of land species and 96% of marine species due to massive volcanic eruptions releasing greenhouse gases. Current human activities like fossil fuel usage appear to be causing equally abrupt and drastic environmental changes as past mass extinction events. Fossil fuels represent carbon that was trapped over millions of years but is now being released in just a few hundred years through human usage. This rapid climate change may be too fast for many species to adapt to. Continued fossil fuel usage and resulting global warming increases the risk of crossing a tipping point where irreversible changes accelerate the climate crisis beyond control. Keeping global warming below 1.5 degrees Celsius is important to avoid such risks, but current nation pledges under the Paris Agreement may still be insufficient to achieve this goal. The passage expresses optimism about technological advancements and their ability to solve environmental problems like climate change. It argues that technology is progressing exponentially, not linearly, meaning the pace of progress is accelerating. Examples are given of exponential growth in fields like computing power, genetic engineering, and artificial intelligence. The key points made are, technological progress happens exponentially, so the future impact could be much greater than what we can currently imagine. Advances in areas like AI, genetics, and computing are already demonstrating exponential growth trajectories. Within a few decades, advancements like artificial superintelligence could enable the development of powerful new technologies capable of halting or reversing environmental damage like climate change. The explosion of progress, once a tipping point of technological development is reached, means future problems may be soluble in ways we cannot conceive today. So in summary, the passage delivers an encouraging message that exponential technological change could empower humanity to stop further climate change through innovations we have not yet dreamed of. Here is a summary of key points. The poem expresses creativity in various domains, like art, music, and literature, which were once thought to require human abilities, but now powerful AI programs can create in these domains as well. ChatGPT wrote a poem in seconds on the topic of human creativity that would have taken the author months to write one as good. If AI continues advancing exponentially, combined with other technological breakthroughs, it may help address environmental issues by improving efficiency, like DeepMind did for Google data centers. However, excessive resource exploitation also threatens humanity's existence, as seen with COVID-19, likely originating from animal to human disease spread due to habitat invasion. Technology also helped address COVID-19 through tools like testing, vaccines in under a year, but future disease risks remain if environmental trends continue. The future is uncertain. Rapid innovation may help overcome environmental crises, or we may face depletion leading to dystopia. The 21st century will be crucial for humanity's long-term future. Our capacity for innovation is a double-edged sword that can lead to both utopias and dystopias, depending on how it is applied. To resolve issues stemming from misuse of innovations, we need to make full use of our innovative abilities to develop technological solutions that can address problems like climate change. We also need to advance our understanding of human psychology and behavior to foster greater global collaboration in solving environmental challenges. A multifaceted approach is needed that utilizes both technological breakthroughs and insights into human nature to effectively address ongoing climate and environmental problems. All available social and technological resources must be leveraged through coordinated global cooperation. Innovation offers hope if guided properly, but poses risks if not managed responsibly. The task involved a rat foraging for water in a tea maze, with the two target locations for receiving water changing their probabilities. Left target was 21%, then 72%. Right target was 63%, then 12%, without any sensory cue. This dynamically changing task aimed to emulate decision-making in an uncertain environment, where precisely tracking the true value probability of each target is challenging. The rat had to keep track of the water delivery probabilities at each target based on its history of past choices and outcomes at each target. Recent experiences would be weighted more than remote experiences. Reinforcement learning models formalize how the rat could estimate the changing values of each target and make choices that balance exploiting the highest valued target with exploring other targets when values may change over time. Recording neurons in the rat hippocampus found that some neurons tracked the changing estimated value of one of the targets, showing they encode value information that could support this kind of dynamic decision-making.